In the name of Allah, the most gracious, the most merciful. Assalamu alaikum. Today we will discuss about sexual reproduction in fungi, methods of sexual reproduction, and at the end, parasexuality. So let's start. The sexual reproduction in fungi involves the fusion of two compatible nuclei. The process of sexual reproduction consists of three distinct phases. Plasmogamy, which involves the fusion of protoplast to bring the nuclei together. What is the protoplast? A cell without its cell wall is called protoplast. Karyogamy involves the fusion of two nuclei followed by the meiosis, which restores the haploidy. Sex organs. The fungal sex organs are called gametangia. If they have similar morphology, they are called isogametangia. If they are different from each other, they are called heterogametangia. In heterogametangia, the male gametangium is called antheridium and the male gametes are called antherozoids. On the other hand, female gametangium is called oogonium and female gametes are called eggs. The compatible nuclei are brought together by any one of the following methods during sexual reproduction. These methods are called methods of sexual reproduction. The first one is the planogametic copulation. In this type of sexual fusion, two motile gametes fuse together and they convert into the zygote. The second one is the gametangial contact. In this type of sexual fusion, two gametangia of opposite sex come in contact with each other, and the male nuclei migrates into the female gametangium. This migration is brought about either by the development of a pore or by the dissolution of wall of contact or by the formation of fertilization tube. The third one is the gametangial copulation. In this type, direct fusion of entire gametangia takes place. It means the entire contents of one gametangium are transferred to other compatible gametangium through a pore. The fourth one is the spermatization. In this type, small uninucleate spore-like male structures called spermatia are produced. The spermatium attaches itself to the trichogyne of female, and wall of contact between the two dissolve. The male nucleus migrates into female gametangium and fuses with the female nucleus to form the zygote nucleus, and this type of sexual fusion is called spermatization. The last one is the somatogamy. Fusion of two somatic hyphae of different species, and specialized sex organs are not produced in this type. The last topic is parasexuality. What is parasexuality? Parasexuality is a unique process in which plasmogamy, karyogamy, and meiosis takes place in a sequence, but not at a specified time and specified points. Normally, the parasexual cycle occurs in those fungi which lack true sexual cycle, just like deuteromycetes. So let's start. This is the complete cycle of uh, parasexuality, or we can say that somatic recombination. Parasexual cycle, also called somatic recombination. This is the first step of parasexual cycle in which two different fungal strains involved. And first step is the cytoplasmic fion and are also called anastomosis. Anastomosis is equal to the plasmogamy in sexual cycle. And this is the fion of cytoplasm between the two hyphae. And next step is the mixed haploid nuclei and produce the production of heterokaryon, and this process is called heterokaryosis. And you can see that this is a single hyphae which contain two different types of nuclei. So that's why they are called heterokaryon. And next step is the production of heterozygous diploids, and in which fusion of two nuclei takes place. As a result, heterozygous diploids are produced. As you people can see here, this is the heterozygous diploids. And the next step is the mitosis, in which heterozygous diploids and the homozygous diploids undergo mitosis, and there are different parental and recombinant genotypes. And the last step is the restoration of haploid state, in which, as a result of mitosis, some produce haploids and some produce aneuploids. Aneuploid is a condition having the abnormal number of chromosomes. So after karyogamy, the nuclei become diploid. But during the mitosis, chromosomes are sometimes lost unevenly. So the diploid nuclei transition to an aneuploid state due to the random loss of one or more chromosomes. But over successive mitotic divisions, the aneuploid nuclei may stabilize or evenly lose enough chromosomes to become haploid. So what is the what are the aneuploids, or what are the benefits of aneuploids? Aneuploidy introduces basically novel combination of genetic traits that may result in new phenotypes. Aneuploid fungi can sometimes better withstand environmental stress, as their genome variations may offer survival advantages. And also, aneuploids acts as an intermediate step before haplodization, ensuring the creation of genetic variants. And these are the two significant uh, important features of the parasexuality. They provide the genetic variation and also help the fungi to survive in the changing environment. What is the difference between parasexuality and sexual cycle? These are the key differences. First one is the recombination occur in the mitosis in the parasexuality, but in meiosis during the sexual cycle. Gametes are not involved in parasexuality, but the gametes involved in sexual cycle. And last one is the Chromosome aggregation is irregular in parasexuality but regular in a sexual cycle because meiosis ensure the proper segregation of the chromosomes.